We usually think about food through the lens of chemistry. We count calories, track vitamins, and monitor how carbohydrates break down into circulating glucose. Mechanical forces dictate how we process nutrition. Our internal biology functions as a structural machine alongside its chemical reactions. Take the extracellular matrix, or ECM. This structural network of proteins, like collagen and elastin, suspends our cells in a flexible web. In a healthy state, it acts as a biological shock absorber, flexing to distribute physical pressure before bouncing back into place. Maintaining the physical stability of this scaffolding is as critical to our survival as keeping our blood sugar balanced. A high-fat diet physically hardens the tissue surrounding fat cells, moving beyond the simple expansion of cell size. The local architecture changes. Structural collagen thickens, and flexible elastin degrades, while advanced glycation end products, or AGEs, cross-link the tissue, binding proteins tightly together. This flexible web becomes a rigid cage, losing its elasticity and actively compressing the trapped fat cells. To understand how this stiffening causes diabetes, we must investigate the body as a collapsing architectural structure. If we zoom down to a single fat cell, or adipocyte, we can see it is now completely trapped inside this hardened scaffolding. The cell recognizes its environment has hardened through a process called cellular mechanosensing, where it literally feels the stiffness of its surroundings. The cell membrane is studded with integrins. These proteins act like physical tent pegs, linking the outside matrix to the cell's internal framework. As the matrix stiffens, those outside fibers lose their slack, violently pulling on the integrin pegs and creating high mechanical tension. Physical tethers anchor every cell to its surroundings. When the environment turns rigid, it forces immediate mechanical stress onto the cell's internal structure. This tension travels according to the principle of tensegrity, or tensional integrity. Architect Buckminster Fuller popularized this building principle, where structures stabilize themselves through a balance of continuous tension and compression. Tensegrity relies on opposing forces. Rigid struts resist being crushed, while continuous elastic bands provide tension, creating a stable, self-supporting geometry. Our cells use this exact architecture. Integrins physically tie to the cell's internal cytoskeleton, a web of filaments giving the cell its shape. A pull on the surface travels down this tensegrity structure, faster than a chemical signal, straight into the nucleus, warping its shape. This physical distortion triggers mechanotransduction, where the cell converts physical force into biochemical signals, changing its gene expression. The rigid environment pulls on the cell's internal scaffolding, rewiring its communication system and dictating how the cell functions. That constant physical squeezing eventually causes the cell's internal machinery to malfunction. This mechanical stress jams the cell's internal signaling by increasing the phosphorylation of IRS1. IRS1 is a signaling protein that acts as an internal switch for insulin processing. That jammed signal reduces the number of GLUT4 transporters at the cell surface. GLUT4 is a protein moving glucose from the bloodstream into the cell. The cell removes its own doorways. It becomes physically incapable of absorbing glucose, leaving sugar to accumulate in the bloodstream. This mechanical blockade marks the start of insulin resistance. It is a structural failure that overrides the cell's ability to respond to insulin. For decades, we have treated insulin resistance as a chemical overload. We attempt to fix a chemical imbalance by prescribing chemical solutions. Treating metabolic diseases more effectively requires a shift toward addressing our physics. Exercise and physical therapies are powerful because they initiate mechanical remodeling at the cellular level. Applying these physical inputs alters the tension across our tissues. Over time, that can help restore the flexible state of the extracellular matrix, reversing the rigid damage caused by a poor diet. Every bite of food and every movement we make is an architectural decision. We are constantly building or breaking our own cellular foundation.